Now, Chapter 4 of Class 12 Biology NCERT, that is Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offsprings identical to their parents? Or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with inheritance as well as the variation of characters from parents to offsprings. Inheritance is the process by which the characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well known Indian breeds. Example is Sahiwal cows in Punjab. So we must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of this phenomena. So basically, the scientific basis of this inheritance and variation was not known. So Mendel's laws of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, that is from 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plants, yellow or green seeds. So in this diagram, you can get all the 7 characters that Mendel used in his hybridization experiments. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. So, in next paragraph it's written, Mendel conducted such artificial pollination or cross pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs, which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated or constricted seed, green or yellow pods, and tall or dwarf plants. So just learn this all. So these are the dominant ones and these are the recessive traits. So inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed a tall and dwarf plant, pea plants, to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial 1 progeny or the F1 generation. So Mendel observed that all the F1 progeny plants were tall like one of its parents. None of them were dwarf. He made similar observations for the plants. For the other pairs of traits, he found that the F1 always resembled either of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offsprings were either tall or dwarf. None of them were in between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while the F2 stage, both the traits were expressed in the proportion 3:1. So the contrasting traits did not show any blending at either F1 or F2. So this is about monohybrid cross. In monohybrid cross, there was no blending of the traits. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parent to offspring, through the gametes over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t for the dwarf. And T are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, small t, or small t, small t. So Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for heights are identical or homozygous. That is, both the alleles are of same types. Means both are either capital T or both are small t, respectively. Capital T and small t are called the genotype of the plant, while the descriptive terms means this appears to be tall and this appears to be dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype T, capital T and small t? As Mendel found that phenotype of the F1 heterozygote, that is capital T and small t, to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, that is, as in the F1, and hence is called the dominant factor while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other characters or traits or pairs that he studied. It is convenient and logical to use the capital and lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small d for dwarf because you will find it difficult to remember whether T and D are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygotes capital D and small t, or can be dissimilar as in the case of heterozygote means this one. Since the capital T small t plant is heterozygous for genes controlling one character means height, it is a monohybrid cross. It is monohybrid, and the cross between capital T and this both are representing tall character. Means height, not tall. This is tall, this is dwarf. So this character is known as height. So it is a monohybrid cross. Crossing between these two means monohybrid cross. For the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossing. In this way, the gametes of the tall capital T plants have the alleles T, and the gametes of the dwarf T plants have the allele small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and the small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T and small t allele. Since these hybrids contain alleles which express the contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in figure 4.4. So this is a Punnett square. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all the possible genotypes of offspring in a genetic cross. So remember that it is genotype, not the phenotype. Once you know the genotype, then you will automatically know what the phenotype will be. The possible gametes are written on the two sides, usually the top row and left columns. All the possible combinations are represented in boxes below in the squares, which generates a square output form. The Punnett square shows the parental tall TT male and dwarf TT female. The gametes produced by them and the F1 TT progeny. The F1 plants of genotype TT are self-pollinated. The symbol this and this are used to denote the male, and the male means denote the female eggs and the male pollen of the F1 generation respectively. The F1 plant of the genotype TT where self-pollinated produces gametes of the genotype TT in equal proportion. When fertilization takes place, the pollen grains of tea have a 50% chance to pollinate eggs of the genotype T, as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance to pollinate eggs of genotype capital T, as well as genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes. These are the three types. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to capital T, 1/2 lead to capital T small t, and again 1/4 to small t. Means 1/4 is for homozygous, and another and 1/2 means half of the progeny is homozygous and the other half is heterozygous. Though the F1 have a genotype of capital T small t, but the phenotypic character still seen is tall. The at F2, 3/4 of the plants are tall, where some of them are capital T, while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotypes TT and TT because they both are tall. Hence, with the genotypically, you can just differentiate it because one is capital T small t and the other is small t and capital T. And here only one character is dominant, capital T is expressed. Small t is not expressed because capital T is dominant over small t. Hence, the character capital T or tall is said to dominate over the other allele T or dwarf character. It is just due to this dominance of one character over the other that all the F1 are tall, though the genotype is TT. And in the F2, 3/4 of the plants are tall, though genotypically 1/2 are capital T small t and only 1/4 are capital T. This leads to phenotypic ratio of 3/4 tall and 1/4 small t. That is 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4 is to 1/2 and 1:1/4 ratio of TT is capital T small t and small t is mathematically considerable to the form of the binomial expression ax plus by whole square that has the gametes bearing genes T or T in equal frequency of half. The expression is expanded as given below. a प्लस बी का होल स्क्वायर वाला फॉर्मूला होता था ना, जिसमें a स्क्वायर प्लस बी स्क्वायर प्लस 2ab होता था. दिस इज द सेम थिंग, हाफ टी प्लस हाफ टी का होल स्क्वायर. सो हाफ टी प्लस हाफ टी इंटू हाफ t plus half t. We can write it this way, or we can write it as a square plus b square plus 2ab. Okay, so Mendel self-pollinated the plants, F2 plants, and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. So dwarf means homozygous dwarf, right? Homozygous small t. So this, when you self-pollinate in F3 and F4 generation, is just going to produce all the progeny are going to be what? Simply dwarf. Okay. So he concluded that the genotype of the dwarfs are homozygous TT. What do you think he would have got had he self-pollinated a tall F2 plant? So tall F2 plant. This is a tall F2 plant. This is also a tall F2 plant. So either you are going to self-pollinate this, then you will receive what? All the plants will be tall. And if you are going to do this, then this is just the same case of crossing over of the progeny of the F1 generation, right? So from the preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has T or small t capital T composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called as the test cross. So what it is written now, let me just explain. Whether a tall plant from F1 or F2 has TT or TT composition. To know the genotype, okay, we are not able to understand what the genotype is. So therefore to determine the genotype of a tall plant at F2, okay, we have two tall plants at F2. These are the two genotypes. We don't know what the genotype is. What Mendel did, Mendel just crossed the tall plant from F2 with a dwarf plant. You take one of these plants, you take the plant and cross it with this dwarf plant, and this he called as a test cross. In a typical test cross, what happens? An organism P plant here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. We're not self-crossing it. What are we doing? We're taking the dominant phenotype. Dominant. We don't mean whether it is homozygous dominant or heterozygous dominant. We just need a dominant character, and we're not selfing it. We're crossing them with the recessive parent. So the progenies of such a cross can easily be analyzed to predict the genotype of the test organism. So the progeny that will be formed will help us to analyze the genotype of the organism that we want to test. So figure 4.5 shows the result of typical test cross where violet color flower W is dominant over white color flower small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition of a test cross? So basically this is a test cross. Here what we can see here, that this is homozygous recessive and this is homozygous recessive, and these are the two dominant traits, dominant phenotype. We are just crossing it with the recessive wala phenotype. So यहाँ पे results क्या आ रहा है हमारा कि all the flowers are violet. So since all the flowers are violet, so we know that since all the flowers are violets means homozygous dominant था. This is homozygous dominant. यहाँ पर एक भी अगर recessive वाला रहेगा ना, means this is heterozygous dominant. So at least one. Here it is showing 2 because after crossing over the result is this. So one will come. One recessive trait is going to appear in this. So that is why we are able to understand that in this one is a heterozygous violet. So based on his observation on monohybrid crosses, Mendel proposed 2 general rules to consolidate his understanding of inheritance in monohybrid crosses. Today these 2 rules are called the principles or laws of inheritance. The first law or law of dominance and the second law or the law of segregation. So from here we will study in the next video.